Thank you.
Hi. Right. So yeah, going to be talking about the Monarch Foundation products and projects and products. So we're talking about Phenopackets today. And although we're talking about Monarch, Monarch has definitely been driving Phenopackets adoption and uses, but the sort of primary place where Phenopackets have been developed is actually the GA4GH, or the Global Alliance for Genomics and Health. So you can see their logo on the top left there. And I'll be going through a little bit about the history and how everything fits together. So yeah, it's been going on for a little while. But anyway, let me talk me through it. So let's just start off and say, what on earth is a Phenopacket? So you've probably heard the term. A lot of you probably know exactly what it is, but for people who don't know, you have to ask ChatGPT what everything is nowadays, because Google is totally not cool any longer. So Phenopacket is basically a standardized digital container for sharing detailed phenotypic data about individuals, typically used in biomedical research and clinical practice to facilitate the exchange of complex health information. So Phenopacket encapsulates data such as clinical symptoms, medical history, genetic information in a structured interoperable format, allowing for the integration and comparison of phenotypic information across very different studies and platforms. It's most prevalent in fields like genetics, rare disease research, and personalized medicine. So it's derived from the term pheno, relating to phenotypes and the observable characteristics of an organism, and packet, a small standardized container of information. So this, you might think I wrote this, actually I didn't. It was written by ChatGPT and it did a really good job. So Phenopackets are well known to GPTs, and they can produce them and understand them quite well. So you might be wondering, where on earth does a Phenopacket come from before, what is it? Where does it come from? So a little history. Back in 2015-ish, something like that sort of time, Melissa and Chris were sort of talking to each other, and Melissa, in a typical sort of obedient way, kind of went, I just need something to kind of like put a phenotype in a kind of packet or something like that. And then Chris went, how about a Phenopacket? And then that was it. A little bit after that, we got together and then started trying to figure out what on earth Phenopacket really is supposed to look like. So we're well having an idea of like what ought to go into it, but there was quite a lot of quite a lot of scope that we could have gone through. So it took a couple of years to come to something that was mostly usable. And then in 2017, we joined up with the GA4GH and presented it at a hackathon in Toronto. And Monarch being one of the driver projects for the GA4GH, and this is where the Phenopacket as you know it at the moment really kind of took form and started off. And surprisingly, it took quite a long time to try and define exactly what it was supposed to look like, just even though we'd already made a Phenopacket beforehand. So a little bit after that, in 2019, we actually came out with the first product approval for the Phenopackets, and this was really very much rare disease focused. And so we had some initial implementations. So part of the acceptance criteria for the GA4GH for a product there is that actually it's being used in some things in actual real life. So it's a little bit of a chicken and egg, because it's hard to create a standard if you don't know what it looks like, if you can't use it, but you need to eventually kind of it sort of becomes a thing. So really Xamizer was the kind of like one of the main sort of driver use cases for that. And I think I'll be talking, or Damien will be talking about that in another talk in a few months' time. And then Lyrical, which is a similar kind of thing to Xamizer, also produced by Peter. And also then you had the biosamples database, of really one of our very first adopters, and the European Rare Disease Networks. So they were all kind of very much rare disease focused, apart from biosamples, which was a bit of an outlier there, but yeah, it was cool to have some more use cases. So then the pandemic hit, and we all wondered what on earth to do with ourselves. And then we got going on the the second incarnation of the Phenopackets. And really, what we were trying to do there was to expand the scope of the Phenopacket outside of just rare disease and into cancer and more sort of general sort of clinical phenotypes. So we're always looking at sort of phenotyping, but in particular, this one will also not just cancer, but also temporality, which is really important to actually be able to have a sort of time course of how a phenotype might change. And then the big scoop of that is that we managed to also, the GA4GH and ISO had a kind of an agreement to try and co-approved products so that there was less fragmentation within the standards worlds. So this the Phenopacket also got approved as an ISO standard. So this has got the ISO number 4454, then version 2022. So you might be wondering, so how does GA4GH and ISO fit with sort of other bits and pieces? So if we look at, and also how does Monarch fit into all of this? So if we look here, that if you look in the top row here, we've got CDISC, GA4GH, HL7, and Odyssey. So these are kind of these are four sort of fairly standard names in the clinical standards, and they all will collect phenotypes in some way, shape, or form, but they all have slightly different sort of focuses. So CDISC tends to focus more on clinical trials, doesn't really have any kind of genomic data that I know of in it, and it's also quite sort of more heavily drugs focused. GA4GH is really focusing on genomic health. HL7 is the sort of primary means that EHRs interoperate with external software, and then Odyssey is a kind of a clinical data warehousing. So they produce a schema for SQL schema for things. So if we then focus down a little bit more to the GA4GH, the GA4GH has different work streams. So within it, so this is the second row down. So we've got, for example, the genomic knowledge standards, and the GA4GH is great on acronyms as well. So that would be GKS, and then there's ClinPheno for clinical phenotypic data capture. Then there's the large scale genomics, or LSG as we like to call it. So that's things like the VCF files and BAM files, bed files. I think all these kind of large scale file formats for genomics come under the GA4GH under that particular working subgroup. And then there's discovery, and so these are all kind of slightly interrelated. So discovery, ClinPheno, and genomic knowledge standards are all interrelated in one way or another. So discovery houses things like Beacon and Data Connect, and then the genomic knowledge standards has other things like the VRS, which is the variant representation standard, and also MapBrush is working on that one for actual knowledge about what a particular variant or piece of genome would actually sort of any kind of properties that might have. And the ClinPheno is obviously clinical phenotypic data capture. So they're going into the ClinPheno group. We've got subgroups within that, of which Phenopackets is one of them. And then there's a pedigree subgroup, which we're looking obviously pedigrees and family history, and then the pharmacogenomics data standardization. I think there's a couple more, but those are kind of a bit more nascent. So Monarch has got actually got stuff, has input into the GKS, ClinPheno, and discovery to an extent. So we all have to peer review each other's work, and then there's the GA4GH also has a steering committee, which the work stream leads and Delivery projects are also part of. So Monarch's kind of heavily involved within the whole of the GA4GH. But our sort of primary focus, at least from my perspective, is Phenopackets. So then you might be wondering how Phenopackets actually fits with other clinical data. So as I said before, you've got Odyssey and HL7 Fire. So HL7 Fire is another incarnation of a messaging standard. So beforehand they had, I think it was HL7 V2, I think it was called. And then Fire turned up, and now Fire has been through several iterations as well. And so obviously standards on their own are wonderful and they're helpful, but if you have a standard, your data in one standard and you want to have another standard, then you need to have some sort of interoperability. I mean, obviously that's the key part of standards, is that because they're standard, you can hopefully interoperate between them. So we actually have the ability to export data from OMOP, from the Odyssey, from their database into Phenopackets, and also out of Fire using this thing called an implementation guide. Again, from Fire messages into a Phenopacket. This isn't always 100%, so you will lose data in some way, shape or form somewhere, but even within Fire and Odyssey, there's a lot of different ways of doing these things, so you can lose data all the way through. So none of these are 100% perfect in terms of what they will do for interoperability, but it's a start. So you can get the kind of the core of what you're trying to express is pretty well transferable between these systems. So the use cases for Phenopackets, like I sort of say, just going a little bit more sort of into like why do we have them. they are many and various, and this is sort of what one of the images that we had right from sort of earlier on, and I think we've done pretty well in terms of what we expect and how things are actually being used. So I mean, we sort of assumed that originally that the physicians and patients and families would be creating their Phenopackets along with the researchers and or paper authors if they're going to be doing clinical case notes and these kind of things. And I think what was sort of coalesced is that maybe the genetic laboratories and some sort of researching, well, anyway, we've got a lot of different places where they still very much can be used and is used. So you can see these various methods here. So basically, it's sort of coming from a lot of different ways of phenotyping people, and also how they link together with pedigrees. How they can then be coalesced for these and then put into a Phenopacket. And then from the use cases you can get with a Phenopacket is pretty much anything you can think of that would involve some sort of standardization of phenotypic data. So for example, we've got finding out cohorts, stratifying patients, finding other patients that look like other patients, actual diagnosis of the patients for genomic diseases or phenotypes, then trying to sort of translational research I don't think we've done quite as well in. Mainly because the Phenopacket is very much human focused and not so much for model organisms, but and then decision support, which I guess is sort of part of the diagnostics. So clinical labs, with being of our users at sort of the end use, I think they are definitely one of the potential winners here. Electronic health records would be something that would come out of a Phenopacket, but there's nothing to say that you couldn't store Phenopackets within an EHR. Patient registries would be a major win should they do start to use these. Some of them, I think, for the matchmaking part of case would be using these already. Journals, I don't think have actually started to really uptake these, which is a shame, but because that would be really perfect for being able to sort of help with the patient stratification and just generally having a better idea about what really what the phenotypes for a particular disease is. And then databases have definitely uptaken Phenopackets, so there's some fairly good uses there. So within sort of genomic medicine, which is really my sort of my thing, the Phenopacket sort of sits into more into the diagnostic side of things. So if you look at, say, where the clinician and the EHR part is, the EHR and the clinician obviously they kind of interface with each other to an extent, but there's sort of a lot of read out of the EHR stuff gets put into it. But certainly phenotypic data as such as we would think about it from the sort of precision phenotyping perspective is not really the kind of thing that the EHR is particularly good at. Whereas something like phenotypes is absolutely bespoke for, and that's entirely what it is meant to do. So you can get some very, very precise, HPA phenotypes out of phenotypes and actually export that as a Phenopacket. And these kind of things can then be taken in, along with the VCF files and used in bioinformatics pipelines for actually solving cases. But this is the kind of the general workflow that we sort of really envisage that the Phenopacket would be particularly useful for in genomic medicine. So let's go into actually what the Phenopacket looks like. So as I said before, the Phenopacket is a schema, and it's been developed over several iterations. And this is the current iteration, which is the V2. So if you look at if you look at the very top of the diagram, you've got the family and the cohort boxes. So those are two sort of high-level concepts which the Phenopacket itself, which you can see in black, would be part of. So for example, you could have a cohort of Phenopackets, so there would be many people would be grouped together for some reason, whatever you want to be used and defined as the kind of common characteristic for your cohort. And then you can put all the examples of those Phenopackets within the cohort. The family is more of a rare disease informatics sort of usage. So for this one, again, you have a family which would have multiple Phenopackets in there, but the key thing here is that you have a pedigree attached to the family, so you can see how the individuals are related. And then you tend to have a proband, which would be the person who was being investigated for a particular genomic condition. And then you can see how using the pedigree, you can see how the genetics may have informed the phenotype. So from that, if you look at going down, you have an individual, which is part of the Phenopacket, so it's really what the Phenopacket is to do with. diseases, interpretations, biosamples, metadata, medical actions. And then if you look at those, they kind of, you have more information that you can actually add into these things, such as measurements on biosamples, phenotypic features on biosamples, procedures to do with medical actions and treatments. And then on top of that, we've then got the idea of these time elements. Actually, one thing that's not in here, which would be really most important for Monarch, is the concept of an ontology term. And it's kind of like, you can't even see the ontology term because the ontology term is everywhere in this case. So almost every single concept is uses an ontology. So we like to try and tag everything or express all concepts within the Phenopacket as an ontology term of some sort, so that you can then start to reason about things, and you can have, you know, fuzzy matching on things, so you really understand what something is, rather than sort of having to try and guess and look it up or use some sort of lexical matching or something. So if we're moving on from here, we're just going to focus now on the actual Phenopacket part itself. So if you take the Phenopacket in isolation, we're really just looking at an individual. So it seems a little bit weird. You've got an individual which is part of the Phenopacket rather than being the Phenopacket, but it is possible to have, say, a Phenopacket of just a disease with some phenotypic features, maybe just an interpretation on its own, but then it's sort of a little bit awkward to have that in it, or just a biosample. So it's meant to be kind of flexible, but the individual is kind of part of an expression of the Phenopacket and what it is. So say, we're just going to go a quick walk through these, and this is taken from an example paper written by Peter and can't remember who else. Anyway, GF4GH Phenopackets practical introduction, and this is just an idea so that you can get a very good idea about exactly what all the precise fields are in a Phenopacket as you see them, rather than reading the schema itself. So the first idea is that, you have an individual, and you want to be able to describe the individual. Obviously, you want an identifier, you have a date of birth and a sex in where they have, whether they're alive or whether they're dead, what time they died, if that was the case, and also what they died of. For cancer, we've got survival time in days, so this would be the survival time for since they were diagnosed. So you can actually draw some conclusions as to whether any treatments they might have may have actually had a beneficial effect on their disease. So just sort of looking at this, though, there's a couple of things in here. So sex is obviously part of the enumeration. So we've got male, female, unknown, I think. So these are linked to some NCIT terms for sex, but we just define these as any number here because it actually fits better with, say, how fire works, so that works better for interoperability in this case. The date of birth is, again, something which is not required. So what we're trying to do really with the Phenopacket is we're want to make it so that the amount of personally identifiable health information is really limited to the actual usage that you require. So it's quite legal to just have a Phenopacket with just an identifier and really not much else in it. How useful that is dependent, obviously, on who you're sharing it with and the reason for sharing it. So, but that's one of the kind of the primary guiding concepts that we have within the Phenopacket is that most of these things should be optional if it's required to protect the identity of the individual, because all of these things are meant to be used outside of the EHR, where so within an EHR you expect it to be very identifiable because that's the whole point of it. So you want to be there for treating the individual, but outside of it, the use cases can be fairly different. So moving on from the individual, we've got the phenotypic features. Now this is kind of the primary reason for a Phenopacket, obviously given the name. And the phenotypic features themselves, as I said before, are defined in terms of ontology terms. So you can see the ID in the label. This is fairly sort of should be fairly obvious, fairly familiar to you guys. But the thing is, this is really the reason that the Phenopacket, just having a phenotypic feature, just as a list of HB terms, for example, doesn't give you a very, what can you say, a very sort of full-featured or very descriptive idea about what the actual phenotype of the individual is. I mean, if you can have a vague idea about what they might look like, but you don't know really a lot of information, like, give me a little bit more. So clinodactyly, you've got sort of wonky fingers. So which fingers are wonky with? In this case, it's on the right hand. And then also you want to know maybe when the phenotype actually occurred. You want to be able to say whether the phenotype was actually looked for, but then was not observed, so that would be the excluded type. You want to say how severe a phenotype is. You can have other modifiers from the HP modifier ontology, which I can't immediately remember what we might have. Well, that would be laterality in this case, so left or right. So we got the onset, and also you can have these the severities, like as I said before, and also if you're very lucky, you might have a resolution. So rare disease phenotypic features, what happens is they tend to get worse as they get up, as the patient gets older and the condition progresses. Also, they tend to get more phenotypes as they go along, and they don't tend to resolve themselves. If you have something like a cold, then you hope that that would resolve itself eventually, or COVID maybe. So that's how the phenotypic features are worked out. So we if we go back to this just before we move on, you notice that all of these terms are for the HPO. So we've got HPO identifiers all the way through here. We've done that deliberately because we sort of designed the Phenopacket with the HPO in mind, but there's nothing to say that we're not requiring that people actually use the HPO. So it's perfectly possible to use something like SNOMED terms if you wanted to in this one, but they might not always fit particularly well. So so for particular things, we might suggest particular ontologies just to help to keep people in the right place to aid interoperability. But really, with this, it's just meant to be an empty skeleton that you can put whatever types of ontologies or terminologies you need to within it for your own uses or with whoever you're sharing it with. But obviously, the more standardized, the more that people keep their ontologies, the easier it is to share later. So they're moving on with the measurement. So if you had, for example, in this case, this is actually taken from a case with a 6-month-old girl, I think it is, and she had these particular phenotypes, and then the phenotypes, the clinician actually did some measurements on the eye pressure, for example. So you notice here we're using a different ontology or terminology. We're using LOIC, which is for the lab measurements, and then you get a actual value of the measurement that you've taken. In this case, it will tell you what the unit is, what the value of that unit, and what the value of the measurement in that unit is, and also the reference range for the measurement. So in this case, we've got you see at the top here, so you've got quantity, the unit is in millimeters of mercury. So we're using the unit ontology for this one. I think there's a UCUM, which would be a little better to use nowadays, but at the time that wasn't available, at least as an ontology. You see it's 25 millimeters of mercury with a reference range being between 10 and 21. So this is actually a little bit too high. And also you have a time when this was observed. So if you look in here, so the time observed, again, we're trying to give people the ability to express themselves in as precise or loose a way as possible in order to, maybe bin patients into particular ranges, so that again to try and protect their identity, but also so that it's also usable. In this case, we're using an ISO 8601 duration, so you express age in terms of how long someone has been alive for. So in this case, it's the P is for a period, and then 6 for 6, and then N for months. So they were old. They were 6 months old when this observation was taken. And so, yeah, the measurement is really some kind of really quantifiable measurement. So obviously in the HPO, it's more less precisely quantifiable, but obviously we've got things like the LOIC to HPO project where you can actually convert a LOIC term into an HPO term. So in this case, you might have elevated intraocular eye pressure, for example, would be the output of one of these in HPO. And then moving on, so the patient they had their eye pressure taken, and then because of this, they then also thought, well, given the phenotype, we're going to have to actually do a biopsy of the eye. So you see in here, we've got a sample tissue using a Hebron term for eye. And then the phenotypic features of the eye, you can see we're using some NCIT terms here, which is particularly good for cancer phenotypes. And again, we have a measurement. So this is like a little phenotypic, a little phenopacket with all of its own sort of wrapped up in another phenopacket. So the biosample is just a phenopacket almost in its own right. But then because it's really meant for histology, and so we have things like the actual tumor progression that you can put in here. So there was a TNM finding here. So you can see the retinoblastoma, stage 3. Sorry, ETR, I can't remember which that is now. Anyway, it's fairly well advanced, so they decided they had to actually so this is that's fairly well advanced. So it says here that the procedure was actually taken from enucleation of the eye. So the eye was removed, and then when the patient was 8 months old, and also there was some somatic whole genome sequencing happened. So here you see the files here. It gives you your right to the file itself, and then tells you what the genome assembly and the file format was. So obviously, taken from that, they then said, well, okay, we know that the we're going to diagnose the patient with retinoblastoma, and given it a disease stage again. And again, we've got the primary site of where the retinoblastoma was occurring. So we could have sort of 2 different diseases with different primary sites. So all of these things are catered for within the disease category. You can have multiple different diseases. They can have different onsets. They can have different pages of resolution. The disease stages, that would be kind of quite cancer specific in this case. So moving on from that, we had the eye sample was taken, this eye was enucleated, and then a VCF file was taken, and then from this there was a genomic interpretation that happened. So you can see that if you look at this progress status, it says that it was solved. So they've actually found what the cause of the retinoblastoma was. That's given a diagnosis for a disease for the diagnosis. So we know that you have. If you think of, you could have multiple VCF files taken for different reasons. And so you could have retinoblastoma and then some sort of other carcinoma of some variety, and they could each have different genomic interpretations or different causations. So each interpretation would be separate within the phenopacket itself. In this case, we've got a subject or the biosample ID. So the subject was the proband. And then the interpretation of this was that it was actually causative of the retinoblastoma. We've got an ACMG pathogenicity classification. So this is so the ACMG stands for the American College of Medical Genetics, and they have defined a set of criteria for saying whether or not a variant is likely to be causative of a particular condition. So in this case, pathogenic means they're 99% certain that this particular variant is actually causative of the retinoblastoma. So there are other criteria for this: likely pathogenic, which is like a 90% confidence, variant of uncertain significance, which will be anything between nothing and sort of 89%, and then down to likely benign and benign, which are pretty certain that that's really not the case. In this case, there's a therapeutic actionability as well, so you can sometimes take action against particular variants that will then be known to be you could actually solve the case, or not solve the case, cure the case, hopefully. And then the variation descriptor. So this part here is actually part of another GA4GH standard. So this is, I mentioned beforehand, the GKS, the Genomic Knowledge Standards. So the primary standard that they've produced is the variant representation standard, so the VERSE. And this is using part of their VERSE and versatile, I think they call it. There's another way to have, which is how to wrap up the way of the VERSE, the way of actually describing the variant in some sort of extra description would be useful. So in here, we're looking at the VERSE. We've got the variation descriptor, which is like, what is the variant? And its variation, its type of copy number variation. And then you've got the actual location on the reference sequence of the allele, where it starts, where it ends, and then also the copy number, which is only one, so we've got a loss. And also it's a mosaicism, so this is a bit of an extension. So this is something, it's a way of expressing things that aren't actually codified within the standard itself. So that's part of the VERSE spec. So this is where we have Phenopackets and VERSE also part of the GA4GH, so kind of trying to actually utilize each other's standards. So then moving on, we've got from this, we got a diagnosis, and then so now if we know what the problem is, we can then try and solve it. So here we've got some medical actions that are being used, and initially the patient was treated with melphalan, sorry, and then because we want to know really how it was administered, which has got this route of administration. Again, we're using ontology terms for describing these things. We've got drug central ID for the agents, or NCIT for the route of administration. Again, these things could be different. You could use drug bank or KEBI presumably for many of these things as well. We want to know obviously when it was given, how often it was given, and how much was given, and then also what the treatment target was. Like, why are you giving the drugs to this particular patient? So this is what the treatment target is all about. So it's meant to treat the retinoblastoma that they were diagnosed with, and then the intent of the treatment is actually to cure it. The problem is there were some vasospasms, so the patient was not able to tolerate the drug. And then there's a termination reason. So because of this, there was an adverse event. Then the treatment was terminated. So then that was this medical action, the treatment medical action. There's another one, which is a therapeutic regimen, which again, they were treated with carboplatin. So this is some more attempts to actually treat the retinoblastoma. So again, you can see the treatment target was retinoblastoma. The intent was to cure it, and it was unsuccesssul, which is why there was then a procedure taken, which you can see was the enucleation of the left eye. It's performed at 8 weeks because they were unable to cure it using any kind of drugs. And I think that was a successful one. So then if we look at now, so we noticed there was some files in the biosample. So the files can be attached to the biosample itself, or for germline would be into the patient, into the actual Phenopacket. These are meant to be fairly fairly loosely defined, just so that because the file can be used for practically anything. But because we've got a real sort of focus on genomics, we've got this file attributes, which is a kind of a freeform map or list or associative array, whatever you want to call it, for describing the attributes of a file. At least for the genomics part of things, we've got genome assembly and a file format where we've got recommended recommended codes that you can put in there to actually describe what the file is all about. And then lastly, the metadata, which is probably the thing that will be most sort of monarchy about this whole lot. So if you look at each one of these resources here is a little bit like the, oh crikey, I've forgotten what the file name is called. Doesn't matter. Anyway, so it basically looks up, so you can do query expansion and contraction on namespaces for ontology terms. So for example, if you've got the take the NCIT. So we've got the name of the resource of the NCIT thesaurus. You've got a URL, in this case it's an AL file, and a version that would be used for that particular for this particular Phenopacket. Then you've got the namespace prefix, which is what you've got the NCIT colon part, and then the IRI prefix, which is the expansion of the query. So the idea is that you can basically cut them up. So the NCIT colon and then the number. You take the number out of the NCIT part and you plug that into the end of the IRI prefix, will then take you directly to what is the definition within the AL file. I think that's all we need to say about this one. Yeah, we've got a Phenopacket schema version just in case we change the schema, which probably will happen at some point in the future. Hasn't really happened once before. And then also a little bit more about like when it was created, who created it by, who it was created by. So moving on a little bit further, so that's just a description of what the Phenopacket looks like. So this is where it's actually being used. I was just doing this today thinking, oh, I'm not really sure. And I'm pretty pleased actually. There's an awful lot of uptake from very different places where this is actually being used. So if we start at the top, we've got duration tools. So this is like, how do we create and validate a Phenopacket? So we've got Phenotypes, which is produced by Iran Busk's company in Canada, and that's an extremely well used piece of software within the clinical sphere for patient phenotyping. And that's really where a lot of the patients are being actually precision phenotyping, where they're taking those, and then that gets put into some lab order form of some variety to try and for rare disease. So it also takes into account the pedigree. We've got the Phenopacket lab, which Peter's group have been creating. And if you haven't used that, that's a really good one for just creating your own Phenopackets off the bat if you want to just sort of do small scale ones. There's Phenopacket Validator, which is a Java command line and library for validating Phenopackets, and then Python tools, which is a Python version of it. If you go around anti-clockwise, we've got diagnostic tools being used. So Xmiser, as I said, is the sort of the primary use case for the version 1. Lyrical, similar kind of thing. So these both ingest Phenopackets natively and will give you diagnosis based on these and the actual VCF as well. Then there's Ventagene, which from Kai Wang's lab. They will use the Phenopacket for, as you might say, guess from the actual name of this thing, trying to guess the gene from the phenotype, or diagnose the gene from the phenotype. Then GenoPheno benchmarking tools. This is sort of recently just been the most useful thing that we've had for a very long time, I found, which is Phebal, which has been produced here in Monarch by a whole bunch of us, and that has been very, very useful for ingesting Phenopackets and then running benchmarking of things like Xmiser, either against other software tools or itself, or what else we need. Yeah, or itself for various different purposes for trying to actually, really assess any kind of impact that any changes we might have had onto the performance of Xmiser itself. So that's a really crucial part of the Xmiser release, actually, and then it runs off Phenopackets. Onto GPTs, similar kind of stuff. Patient cohorts and stratification. So Peter's been very hard at work with the Phenopacket store. So I think we've got 4,600 Phenopackets that he's curated out of various parts of the literature. So that came out of the original lyrical corpus, and then he's expanded that massively over the time. This is has GRCH38 coordinate variants within it. And I know that he wants to try and use that so that we're actually using Phenopackets to describe patients, that we can then add those together so that we can get a much better idea about how disease phenotypes might work within the HPOA, for example. So within the HPOA, you've got individual phenotypic features, and then each of those will have a number of occurrence out of the cohort. So if we've got the ability to actually really build up larger cohorts of these patients, we can have much more accurate HPO annotations. So this is a cool thing that's in the sort of ongoing, in the works, coming soon kind of thing. And then sort of moving a little bit more out of the Monarch world, we're going into things like matchmaking, I think, and discovery. So the Beacon V2 and Data Connector 2 GA4GH products that are, I think Beacon V2 has been accepted now as a new product but they're using the Phenopacket as basically the data model internally. There's a few changes, but it's almost a one-to-one leftover of the Phenopacket schema itself within Beacon V2. And that's been used for patient matchmaking and also patient discovery. So you can give it a phenotype match, say, have you got any patients like this? And then it'll go off throughout the Beacon network, and you can then find the patients that are similar to the one that you're actually looking for. Data Connect does some similar things, but in a kind of a different way, not so hopefully with that one. Then we've got interoperability tools. These are really important. So we've got the FHIR implementation guide, which Peter's been doing with Melissa's group, and that is crucial for taking data out of the FHIR, out of the EHR, and then turning the FHIR messages into Phenopackets that we can then use external outside of the EHR itself. Then conversely, I talked to you already about the OMOP exporters, like how do we take stuff out of a data warehouse and then put it into individual Phenopackets for more sort of patient-centered approach rather than sort of cohort-based. And then there's the Convert Pheno, which I think came from the Beacon group, where they're doing similar kind of things. They'll take stuff out of OMOP and I think some of the CDISC models and various other things, convert them into the Beacon V2 model, and then export them again into different formats as you want them. So it's kind of using similar kind of schema, BP2 schema based on the Phenopackets schema to then do the to use that as a common model for different different clinical phenotype standards. Then the platforms consortia, it gets really big at this point. So we've got the IROD, which is the, I can't remember, but it's a rare disease consortium in Japan for solving rare disease cases in Japan. Bento, which is a platform used by Genome Canada, the EJPRD, so this is the European Joint Program for Rare Disease. So this is a large consortia of groups within Europe who they were one of the first adopters, so which SolvID, I think, is one of them. They were some of the first adopters of the Phenopacket. And then Australian Genomics has been getting on board with it, and as a Monarch, obviously, we've had a huge amount of usage internally of the Phenopacket. It's been extremely useful. Databases, the biosamples database was the original sort of first database implementation of the Phenopacket, and then from there we've also had the EGA, so that's the European Genome Phenome Archive, have subsequently adopted the Phenopacket as a sort of more of a schema, because I think they had a problem that people had no schema when they wanted to actually deposit data with them, and they couldn't really say, oh, use this, use that, or something else. So I think they've settled on using the Phenopacket as a reasonable way of trying to unify most things that people are trying to submit to the EGA. And that GEM Japan is a biobanking database, and I can't understand a word of what's in there because it's all in Japanese, and I don't think you can even access it in this unless you live in Japan. But yeah, so that's one of the key things. I think with Japan, it's they've actually got a mandate to use any ISO standard if one exists for a particular sort of particular usage. So Phenopackets being an ISO standard means that I think Japan are probably going to be using it, or have to use it a lot more than other people might need to or want to, depending on the usage. So that'd be quite interesting to see that. So I know there's one group which are using Phenopackets and Lyrical, and just basically for their entire stack for diagnosing rare diseases, which is a pretty awesome thing to see for Monarch technologies being used like that. Right, so this is a lot of links. I'm not going to read through them because this is basically just a bunch of links to what I was already showing you. Although there's probably two that I should point out here. The most important things you should look at if you're interested in using Phenopackets. So the first one is the link to the Phenopacket schema documentation. That is the most up-to-date, sort of mostly authoritative way of looking at things. If you fancy paying the ISO to get the ISO version of it, then go to the second link down. They're basically the same thing, but the second one has been ISOfied. So Lindsay Smith from the GF4GH did a huge amount of work to translate what was written down in the Phenopacket schema, read the docs, into a big Word document for ISO. And then there's the Phenopacket schema. So this is the reference implementation and the schema definition. In my view, this should be taken as the source of truth, because that is what will be actually produced when it's coming out of any software, because that's the schema as it's defined in Protobuf, and any mistakes should be addressed at that point there. This is a GitHub repository, so there's any kind of requests you might want for new features or issues that are coming up in the schema should all go there. Yeah, I think that's probably what to stop at this point, because there's a lot of links otherwise. And like I said, I've already talked to you about these already. Yeah, so these are some more of our uses from GF4GH, and these are the sort of people involved at the very top level of the GF4GH data capture workstream. So let's see, quite a lot of Monarch people involved in here. And that's it. Thank you very much for listening. I hope you enjoyed it.